Hello students, welcome to the EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am senior scientist in the division of Animal Biochemistry at National Dairy Research Institute, commonly known as ICAR and DRI, an institute located in Karnal within Green State, Haryana, near Delhi, the capital of India. Today, I'm going to talk about the module Protein Sequence Analysis from the paper Biostatistics and Bioinformatics. Friends, computational protein sequence analysis is extremely useful in planning and conducting experiments in the discipline of biochemistry. In this module, you will download a sequence from database for computing biochemical parameters such as molecular weight, isoelectric point, extinction coefficient, and many more. In addition, prediction of transmembrane topology, including signal peptide for directing protein to endoplasmic reticulum, is very useful in elucidating protein transport. And prediction of transmembrane helix serve to detect anchoring of proteins in the cell membrane. Finding families, domains, and sites gains in significance for assigning function to the protein sequence. Finally, finding repeats and their graphic visualization enables to understand their important functions within the cell. First of all, let us download a sequence from database for computing biochemical parameters. We'll use glycophorin A protein for protein sequence analysis. Glycophorin A is an integral membrane protein of the red blood cells. The N-terminal is present outside the red blood cell membrane and is highly glycosylated. Glycophorin A is important for the function because it is a receptor for influenza virus, Plasmodium falciparum, erythrocyte binding antigen, and appears to be a receptor for hepatitis A virus. The glycophorin is having a signal peptide from position 1 to 19 and is followed by a mature peptide from position 20 to 131, having an extracellular domain of 72 residues, a transmembrane helix of 23 residues, and finally a cytoplasmic domain of 36 residues. The subcellular location is cell membrane. Visit XPASI server and search UniProt KB for glycophorin A human. In the ensuing page, select the second entry P02724 and open the Download tab. Select FASTA format and click Go. The sequence of the glycophorin A will appear in the browser window and you can select and copy to paste it in Notepad for saving as a FASTA file. ProtParam tool allows the computation of various physical and chemical parameters for a given protein. The computed parameters include molecular weight, theoretical isoelectric point, amino acid composition, atomic composition, which are self-explanatory. In addition, extinction coefficient, estimated half-life, instability, mean stability index, aliphatic index, and grand average of hydrophobicity, that is Gravy index, is also calculated. Visit XPASI site for the address shown in the slide and paste 131 amino acids of a mature protein sequence which are highlighted in green. That means exclude signal peptide amino acid residues from 1 to 19 position with click compute parameters button. In the results page, The first section displays general parameters such as user provided sequence, which you provided, number of amino acids, molecular weight, isoelectric point, then the amino acid composition, then the total number of negatively as well as positively charged residues. Biochemical parameters such as molecular weight and isoelectric point are useful for analyzing the resolved proteins on two-dimensional gels. Extinction coefficient indicates how much light a protein absorbs, and this is represented by absorbance at a certain wavelength, and it is useful during protein purification process. Lambert Beer Law defined absorbance as a log ratio of the intensity of incident light to the intensity of the transmitted light. This absorbance is simply given by the product of molar extinction coefficient at a particular wavelength for a particular protein with concentration of the absorbance with concentration of the absorber protein and path length through which the light passes. Through the solution, that is the thickness of the cuvette. For commonly used cuvette of 1 cm path length, the unit of molar absorbance coefficient is per molar per centimeter. It has been shown that the molar absorption coefficient at wavelength 280 for amino acids as chromophore is determined by the amino acid sequence of the protein. For each disulfide bond, molar absorption coefficient of 125 is added. Similarly, for tryptophan residues, molar absorption coefficient of 5500 is added. And finally, for tyrosine, 1490 is added. For the shown protein sequence, prot param reported 2 extinction coefficient. From experimental knowledge, it is established that all pairs of cysteine residues in the present protein are disulfide bonds. Therefore, molar extinction coefficient of 14230 can be used during purification of this protein. For glycophorin A, we find no cysteine, no tryptophan, but 4 tyrosine residues. Therefore, molar extinction coefficient at 280 is equal to 5960 per mole per centimeter. Prot param tool also reported the same. Half-life is the estimated time to reduce the amount of a protein to 1/2 after its synthesis within the cell. This is estimated by prot param in 3 physiological conditions. First is mammalian reticulocytes, second is yeast, and third is E. coli. The estimated half-life for glycophorin A, prot param tool reported for all the 3. Since we know that this protein is present in RBCs, its half-life in human is close to 1.9 hours. The instability index provides an estimate of the stability of the protein in a test tube. Instability index smaller than 40 indicates a stable protein, and value above 40 indicates the protein to be unstable. The estimated instability index for glycophorin A is 57.3. Therefore, prot param tool reported the protein as unstable. The aliphatic index of a protein indicates the thermostability of globular proteins and is calculated from the relative volume occupied by small aliphatic side chains of alanine, valine, isoleucine, and leucine. For glycophorin A, prot param tool reported 85.52. GRAVI, which stands for Grand Average of Hydrophobicity, is average hydrophobicity of a protein sequence, and increasing positive score indicates increase in hydrophobicity. For glycophorin A, PROT-PAM tool reported negative value, showing that it is not a highly hydrophobic protein. Transmembrane segments in proteins are thermodynamically stable three-dimensional structures of the integral membrane proteins. Transmembrane helices are usually 20 amino acids in length. The interior of the membrane bilayer is hydrophobic. Therefore, it is presumed that the amino acids which line the helix surface in the transmembrane helices will have hydrophobic amino acid properties. If there is a continuous stretch of about approximately 20 hydrophobic residues in the protein sequence, then the same is an indication for folding into a transmembrane helix. Consequently, transmembrane helices may be predicted on the basis of hydrophobicity of the amino acids lining the helix. Therefore, prediction of transmembrane helices will reveal that some part of the protein is inside the cell and other is exposed on the cell membrane surface, with these two parts linked together through a transmembrane helix. Phobias is a combined signal peptide and transmembrane topology prediction tool and is available online at Phobias. For glycophorin A, Phobias predicted a signal peptide from 1 to 19 amino acids shown in red curve, followed by extracellular, that is non-cytoplasmic domain from 20 to 91 amino acids shown in blue color, then continuing with a transmembrane domain from 92 to 114 amino acids shown in gray color, and to finally end up inside the red blood cell with cytoplasmic domain from 115 to 150 amino acids shown in green color. Top red, which is also used for prediction of membrane proteins, is based on hydrophobicity values for a given size of amino acids window. This prediction shows that glycophorin A is an integral membrane protein. TM-HMM is used to predict transmembrane helices in proteins. For the sequence of glycophorin A, it predicted two transmembrane helices. The first is the signal peptide and the second is the membrane anchor helix. A protein family is a group of proteins which descend from a common ancestor, and therefore these are evolutionary related. Consequently, these proteins belong to a family and have similar sequences, structures, and consequently similar function. Therefore, if a given sequence belongs to a particular family, then it is most likely to have similar three-dimensional domain structure and functional sites. Consequently, the prediction of protein family domains and sites reveal the function of the protein sequence in hand. Interpro Scan, available at EBI, compares the input sequence with protein sequence signature databases to find family, domains, and sites in the input protein sequence. EF0710 protein in Enterococcus faecalis is known to contain domains and sites, as well as it belongs to a protein family. Therefore, download EF0710 protein sequence from Enterococcus faecalis genome browser at UCSC and paste the sequence in the input text box of Interpro Scan as shown next and click Search button. The ensuing results window shows that this EF0710 protein belongs to a protein family and has domains as well as sites. The name of the protein family is phosphoenol pyruvate utilizing enzyme. There are 3 domains and 2 sites in this protein. The first domain is phosphotransferase system N-terminal. The second domain is PEP utilizing enzyme mobile terminal. The last domain is pyruvate phosphoenol pyruvate kinase like domain. There are 2 sites, PEP utilizing enzyme active site with another PEP utilizing enzyme conserved sites. User can look at the details of these domains by clicking on the links provided on the right side. Finding repetitive sequences and their graphic visualization in the protein sequence is very important for understanding functions of the repeat sequences present in the protein sequence. It reveals interactions with cell membrane and cell wall. To understand repeats prediction, one needs to have a protein sequence having many repeats. A cell wall surface anchor family protein in Enterococcus faecalis is known to possess or contain 17 repeats. An online tool, radar, is able to find repeats in the protein sequences, and these repeats can then be visualized using graphical interface dot plot provided by Dotter software. Using cell surface anchor family protein as search term, find all the proteins using Enterococcus faecalis genome browser at UCSC. In the results window, then click on last record in the list for gene number EF3314 and download the protein sequence in FASTA format. Radar available at EBI finds and aligns repeats in protein sequences. Paste the downloaded sequence of cell wall surface anchor family protein EF3314 from Enterococcus faecalis in the input text box and click Submit button. The results page shows that there are 17 repeats of length 88 and 2 small repeats of length 15. The most common sequence feature is the presence of direct repeats in a sequence. Repeated sequences are also called repetitive elements and are most common patterns of nucleic acids and proteins that occur in multiple copies of the genome. The presence of repetitive sequences within a single sequence cannot be appreciated while reading the sequence. Dot plot, on the other hand, is very useful to appreciate the sequence features graphically. Dot plot gives a visual presentation of the sequence repeats. For example, the protein sequence, this repeat is repeated, can be analyzed using a dot plot. To construct a dot plot, one needs to develop a matrix of columns and rows. The number of rows and columns depend on the number of residues in the sequence. In the present case, there are 20 residues. Therefore, a table with 21 rows and 21 columns is drawn. Then the residue symbols for the given sequence are written in the first row and first column. Then the residues are matched for each of the boxes. In the boxes where the residues are same, their symbol is written. Any other letter such as star or a dot may also be written in place of the residue symbol. Then the visual presentation reveals that there is one main diagonal showing the identity. Since we have used the same sequence as horizontal sequence and vertical sequence, therefore there is a complete identity diagonal. The lines parallel to the main identity diagonal in this type of intra-sequence comparison reveals the presence of direct repeat sequences. In the present case, this sequence has easy repeat residues and is directly repeating only once. Therefore, there is one parallel diagonal on each side of the main identity diagonal. Sometimes palindromic sequences such as radar may be present. These are visible as perpendiculars cutting the main diagonal or the parallel diagonal. The sequence EPE is present in this case. In DNA, such sequences represents download dot plot software named Daughter, a dot plot program. The Daughter gives its output, which is graphic and easy to visualize the repeats in the sequence. Say protein sequence of the cell wall surface anchor protein sequence in FASTA format in the same folder in which you downloaded Daughter. To use Daughter, run Daughter on the DOS prompt by typing Daughter, then space, the name of the file, that is 3314protein.fa, then space, then again the name of the file 3314protein.fa, and press enter. The direct repeats are visible as parallel lines. You can count the number of parallel lines. The number of parallel lines shows that how many repeats are there. In the present case, there are 17 repeats. Dear students, in this module, you downloaded glycophorin A protein sequence, which is an integral cell membrane protein of the red blood cells. It acts as a receptor for certain viruses. You learned in silico estimation of its biochemical parameters such as molecular weight, isoelectric point computation, extinction coefficient calculation, half-life, as well as stability, aliphatic, and gravity indices. These biochemical parameters are very useful for understanding the structure and the function of the proteins. Therefore, biochemical parameters can be computed using ProtParam tool at XPazi server for any protein. You also learned prediction of transmembrane topology for detection of signal sequence, which is required for the transport of proteins into the endoplasmic reticulum. And you also detected the presence of transmembrane helices required to anchor integrated membrane proteins in the lipid bilayer. These are the anchor points which dictates which part of the protein will be inside the cell and which part of the protein will be outside the cell. Then you learned to find the protein family, domains, and size for a particular sequence to have an idea about the function of that protein. Finally, you downloaded cell wall surface anchor protein for prediction of repetitive elements and visualized the repeats using the dot plot. These repeat sequences are very much important. For example, in enchirene. Therefore, prediction of the repeats and their visualization adds to the understanding of the structure and function of the protein. Thanks for visiting EPG Pathshala.